I'll begin. Good morning, everyone. You're very welcome to the State of Science webinar that is being organised by the New Zealand Agricultural Greenhouse Gas Research Centre. My name is Sinead Leahy. I work here at the Centre in Palmerston North, and I'll be helping to facilitate this session this morning. And it's a great session. We're really delighted to have both Dr. Sam McNally and Dr. Mike Beare from Plant and Food Research. And they're going to be talking about the work that they've been doing through the centre in relation to soil carbon. Soil carbon, of course, is a really hot topic in New Zealand. So I, for one, am really looking forward to hearing the presentation today. So how the webinar will work is that we'll hear from Mike Beare just introducing the project, and then we'll be followed by a presentation by Dr. Sam McNally. After Sam's presentation, we'll bring Mike and Sam back on screen, and we'll have a little bit of a Q&A with the researchers. So if you look to the bottom of your screen, you should see a Q&A panel. If you open that, you can write in your questions that you would like to ask to the researchers. If you like a particular question, hit the upvote, which lets us know that it's one that you really would like to have answered online. Feel free to write your questions at any point throughout the presentation. So with that, I'm now going to hand over to Dr. Mike Beare. We're delighted you can be here, Mike, and looking forward to the presentation and discussion afterwards.
Thank you very much, Sinead. Thank you very much. Thanks for that introduction, and welcome everyone to the webinar. Look, it's a real pleasure for Sam and I to be able to talk to you today about a topic that we've been working on for several years now, and I think the work continues, and we continue to see opportunities to apply the knowledge that we've gained to practical applications that we believe will ultimately benefit New Zealand's greenhouse gas objectives and also objectives of our farmers. So some years ago, we were challenged by the Greenhouse Gas Centre to answer the question, how much carbon can New Zealand soils store? And initially we were a bit daunted by that question, but we set out by collecting quite a lot of data from across long-term pasture sites in New Zealand, and began with an analysis of that data to see if we could find evidence that would help us to address that particular question. So we started with that sort of analysis to try to determine whether there was an indication of an upper limit of soil carbon storage in our soils. And from there, we then set about trying to test or verify those initial assessments using some more targeted experimental approaches, which Sam will talk about. And then more recently, we've moved on to applying the knowledge that we've gained from that research to try to develop some practical applications of that particular science, which would allow us to look at spatial variability in soil carbon storage potential at the farm scale, and see whether that could help us to inform management decisions into the future. So with that brief introduction, I'll turn it over to Sam now and ask Sam to give our presentation, and then we'll look forward to answering your questions after that. Thank you.
Thanks, Mike. I'll just start off by sharing my screen. So can everyone see that? Can you see that, Mike?
Yep.
Yep.
Okay, cool. So just going to give the presentation about the soil carbon sequestration knowledge that Mike kind of gave the introduction to. And this is really around improving our theoretical knowledge with the intention of trying to apply it for practical applications. First off, I'd like to just acknowledge the many co-authors and colleagues who have been contributing to this work. It's been a team effort, so not just certain individuals, it's been a large chunk of work done by many people. So just to give a bit of a, was it not working? Just why we're interested in soil carbon. first off, soils hold large amounts of carbon, not just in New Zealand, but globally. And if we look at the global carbon cycle, we can see that it's a fairly dynamic process, but soil carbon's in the centre there, in the yellow circle, and it's, I guess the main thing to note here is that soil carbon holds large amounts of carbon relative to both plant biomass and atmosphere. So we've got about 2 to 3 times the amount of carbon in soil carbon globally than what's held in the atmosphere, and about 3 times or slightly higher than what's held in plant biomass alone. So in the terrestrial carbon environment, the soil carbon pool is quite a large pool, and it can be it can feed back to the climate, to the atmosphere, sorry, and either be a source or a sink to atmospheric carbon. So for it to be a source, we basically have microbial respiration and decomposition returning the soil carbon back to the atmosphere, but we can reverse that by increasing plant inputs through photosynthesis and hold it in the soil. So because of this, it can form part of our climate change mitigation processes, and so it is a very important process. I guess acknowledging that this ocean cycle does have large amounts of carbon as well, but in this context, we're just focusing on the terrestrial carbon cycle. So if we can increase inputs through photosynthesis and reduce our outputs through respiration, we can increase our soil carbon pool. But I guess really increasing soil carbon should be a priority regardless of greenhouse gas mitigation. So at the moment, or recently, it's had a lot of attention based on the mitigation potential. And this is considered to be a negative emission. So if we increase soil carbon, it's a negative emission. So we take carbon out of the atmosphere and store it in the soil. And this is, in recent years, so the last decade, it's received quite a lot of attention for this purposes. But soil carbon is involved in many soil functions and is an indicator of soil health. So really this should be a priority for many land managers and farmers essentially. If you have high levels of soil carbon, you'll ensure that your soil is healthy and supports many functions. So just to look at this schematically, here we've got a range of ecosystem services and functions that soil provides. We can directly link soil carbon to a number of these. So the obvious one here is carbon sequestration. So if we take carbon out of the atmosphere and store it in the soil, that's carbon sequestration. And by doing this, you'd have to increase the inputs of carbon relative to the outputs through respiration. So this means that soil carbon can be part of the mitigation solution to mitigate this atmospheric CO2. But also soil carbon is really important for things like nutrient cycling. So soil organic matter, if you have a large pool of soil organic matter there, or soil carbon, you actually reduce the demand for additional nutrient supply. And associated with this, if you're not reliant on as many external inputs, the associated emissions with those also decrease. So we also can build soil resilience by having large amounts of soil carbon. Other things that are really important, soil carbon is linked to things like soil structure. So if you have good soil structure, you would have better water infiltration, better water storage, and those sort of things mean that your things like flood regulation or erosion loss would be improved through this the increased soil carbon. But also things like your soil microorganisms, the carbon actually is a food source for many of these. So if you can have large amounts of carbon, you actually support a really healthy soil that is functioning really well. So that's where increasing carbon, not only from a mitigation perspective, but for these other range of ecosystem services and functions should be a priority. In New Zealand, we're in the fortunate position that our soils have quite high carbon stocks anyway. And this really poses the question, as Mike alluded to at the start, is how much more carbon can be stored? Is there an upper limit? And if where are our soil stocks in relation to the upper limit? Acknowledging that our soils already have high levels of carbon anyway. So a few years back, Landcare led this project where they showed the distribution of carbon stock across New Zealand. And this here is a figure showing the soil carbon stocks for the top 30 centimetres. And on the far right of the screen, we can see that the actual carbon stock, we've got quite a range of carbon stock available. And I guess the thing that sticks out here is that the carbon distribution is quite regionally or or spatially specific, depending on different soil types and where you are in New Zealand. So generally our soils that are developed under recent volcanic activity, such as around Taranaki or Waikato, these have typically got high amounts of elephanic soils. They generally have higher stocks of carbon compared to other soils, which say recent soils which have lower stocks of carbon. So where you are in New Zealand really determines what the stock of carbon is. And I guess this question there is, what is the potential for more carbon storage based on where you are in New Zealand? And acknowledging that we already have a range of carbon stocks already, so what's this upper limit? I guess, why is this important? If we know what this maximum amount of carbon that soils can hold, we can identify soils that have the greatest ability to store additional carbon. And so this is important for on-farm applications such as targeted management. So if you can identify soils that have the greatest capacity to store carbon, you can actually target your approach to, or your mitigation practices to those soils that will respond the best. On the other side of that, if you know soils that are close to the upper limit or close to their capacity, so have a fuller bucket essentially, we can minimize the losses associated with those soils. Another aspect is knowing this is really important for things like policy development, which will help guide things like land use suitability, also mitigation practices. But I think a really important one here is it's also really important for research. So if we want to test mitigation practices that increase soil carbon, we actually don't want to test them on soils that are at their capacity already, because the potential to gain carbon from there is limited. So if we know soils that have the greatest capacity to store additional carbon, we can actually use mitigation practices and test them on those soils to test the effectiveness of those. With this in mind, we know that soil carbon does saturate. There's some quite good evidence in the international literature. This figure here, or two figures, is very recent work that was published last year in Nature Geoscience by Francesca Quattrofo from she's a scientist in the States, and this is some data out of Europe where they've essentially showed on the left-hand figure, we've got mineral-associated organic matter, so that's on the y-axis, with total soil organic carbon, and the figure on the right is particulate organic matter on the y-axis and soil organic carbon on the x-axis. So essentially, the combination of these two pools of carbon, which are measured pools that we can measure in laboratory environment, they would sum to give you your total carbon. The mineral-associated organic matter on the left-hand side, this does seem to saturate or have a point where any additional carbon inputs going in, you don't get any more carbon into that fraction, whereas the POM fraction will continue to increase, which is the one on the right. And so essentially the POM fraction would be regarded as your more dynamic fraction, the fraction that's vulnerable to carbon loss through changes in management, and the mineral-associated fraction would be the longer lived or more long-term lived carbon in your soil. And so if we zoom on here, this is the mineral-associated carbon. It is regarded as the more stable and protected fraction, so it's the part of carbon in the soil which should be the longer lived. But because it's mineral-associated carbon, it does have a finite capacity, so it would get to a point where it saturates. And in this regard, mineralogy of your soil is very important. So with this in mind, and coming back to the New Zealand context, because we know we have a distribution of soil types across New Zealand, we would expect to see differences in our soil carbon stocks. And following on from this, because the mineralogy and your parent material is very important in defining the mineralogy, you'd expect to also probably have a maximum amount of soil carbon in relation to soil types. And a good example here is thinking of a comparison between an elephanic soil and maybe a recent soil. So it's probably unrealistic to think that a recent soil could hold as much carbon as an elephanic soil, and that's just based on mineralogy and what the soils are. So generally our elephanic soils have really high stocks of carbon, and recent soils probably lower stocks of carbon. Some very early work looked at some early work back into the late 90s, early 2000s showed that there was a good relationship between clay content, or also the silt content of soil, and carbon content, and this was particularly done internationally. And so early work that we did looked at this relationship, and in the New Zealand context, the clay content or silt content was a very poor descriptor of how much carbon your soil could hold. So that means that it's not just the soil type that determines your capacity. There's other things going on as well. So because of that, it led us on to look at, as part of defining this upper limit, we wanted to look at what are some of the key properties that would define how much carbon your soil could hold. And in New Zealand, the key thing that stuck out was the mineral surface area was the biggest factor. So the soils that had high surface area had a greater capacity to hold carbon. And this really fits for those aliphanet soils. So aliphanet soils have really high surfaces or surface area, and that means that they can naturally hold a lot of carbon. So to define the upper limit of soils, what we did is we took a range of measured soils across New Zealand, mainly out of long-term pastures. And this was because long-term pastures in New Zealand were considered to be the closest to the upper limit. And so it was a good choice of land use to test this method. So with this measured data, what we did is we applied quantile regression. And with this, we set a 90th quantile, and that was what we said, this is what the upper limit of these soils could hold. And so this figure here, on the y-axis, we've got the fine fraction carbon concentration, which is essentially a mineral associated carbon. And on the x-axis, we've got specific surface area. And you can see the hollow circles are the actual measured data, and the fitted or filled in circles are the predicted data using that quantile regression, and the solid line is the actual quantile regression model there. And so essentially in this regard, what we'd say is the line that we see on this graph is the upper limit, and the distance from each individual hollow circle to that line would be the potential for that individual soil to store more carbon. And so this was work that was done between sort of 2000 or early 2000 or late 2010 through to about 2017 where we published this work. And so this work we actually defined for a range of soil orders across New Zealand and looked at the sequestration potential based on the areas under long-term pasture of these. But I guess the question is, what is achievable? So what we did here is we defined the upper limit, and that's sort of saying this is our maximum sequestration potential we could have based on this upper limit. But then there's a question of what's achievable. So this figure here shows the box essentially is just soil carbon sequestration potential. On the far left-hand side is your maximum biophysical potential. And as you move to the right, it's essentially limitations which reduce that potential, what you can realize. So you have a limitation of what's physically achievable. And that might be the size of your carbon molecules are too big to access some of the surfaces on your soil. So you've actually got a physical barrier there that prevents that sequestration potential being realized. You might have economic barriers. So the cost of doing these practices is too much, so you can't realize it. And then there's a whole range of things which reduce your potential. And so what we really were interested in, we think that what our upper limit that we defined in the early work which I just covered was this physically achievable based on surface area. And we really wanted to look at what's achievable. So it's okay to have this theoretical upper limit, but can we look at what the potential is relative to that? And so that kind of really drove some of the more recent work that we'll cover soon. Just to recover this figure, when I talk about deficit, what I want to sort of reiterate is the deficit that we talk about is the difference between the upper limit that's defined and the current carbon content of a single soil. And so with that, you can get a deficit and work out the sequestration potential of that carbon in that soil. So when I talk about deficit, that's really what I refer to as, so in this instance, the red arrow would be the deficit of the circle at the bottom of the arrow, the size of that deficit. So essentially the work recently, what we've tried to do is we've tried to independently test this deficit model. And to do this, we did two different experimental situations. One, we had a 13C labeled residue addition. So this is a laboratory incubation. And this is really a microbial mediated process where you're adding carbon to a soil, you're letting it, you're incubating it in controlled conditions and letting basically the microbes process that material, and then you're looking at the response of that addition to your soil carbon. The second one was a DOC adsorption experiment. So this one was where we take carbon that we've extracted from a soil, we add it to another soil and we shake it up, and we're looking at the short-term adsorption of that carbon onto the surfaces of the soil that we shook it in with. So that's a really short term, and it's directly the interaction of the mineral surfaces with that carbon molecule. So there's two independent methods to try and test this deficit model, where sort of one was going through a microbial pathway, the other was just looking at the interaction between the carbon and the surfaces. And I'll talk about both those methods in detail soon. Another aspect of this work was we really wanted to take this sort of theoretical deficit model and try and apply it at the farm scale. So up to date, all the work that we've done was quite a sort of national scale, looking at soil orders, and it wasn't at the farm scale. And so we really wanted to say, could you take this information that we've got, we know, and can we apply it at the farm scale? And does that mean we can actually have targeted mitigation practices or highlight vulnerable areas to basically avoid losses of carbon from those areas? And the motivation here was really about translating this theoretical knowledge into a tool that can help inform management or can add value to other monitoring programs. And so Paul Mudge from Landcare is leading a monitoring program looking at change in soil carbon with different land uses, but also he's led a report about how at the farm scale you might measure carbon stocks on your farm scale. And so really for us it was like, how could we add value to other programs like that, where this knowledge could help a farmer basically identify where change would be likely or possible. And I guess if you know that information, you could target some mitigation to try and realise that soil carbon change. So for the incubation experiment, basically this graph here, what I've got shown is I'm showing on the y-axis is the steady state respiration. So this is what the microbes are respiring. And this refers to the carbon input that we added, not the soil carbon, so only the carbon input that we've added. And on the x-axis, that's in relation to specific surface area of the soil. So we had a range of different soils with different surface areas. We added the same amount of carbon to these soils, and then we looked at the respiration associated with that residue. And what we saw here was that the soils that had the higher surface area actually respired less of the newly added carbon than soils that had a low surface area. And so this red arrow just really shows that trend. There was only relatively few soils just on basically the setup we could do and what we could handle. But the general trend here is it looks like that the increasing specific surface area of the soil helps protect newly added carbon. And so if this is the case, that means that if you know soils that have high, across your farm or whatever, or land area, if you know soils that had higher surface areas, they're likely to respond to increasing carbon input by being able to protect more of that carbon. Additionally, what we did is in this incubation experiment too, we had two different rates of carbon input. So here, the idea was to test, if you increase carbon input, do you get a response of the soil that has more carbon input being added to it? And does this relate to deficit? So if I show here if the green rectangle represents the carbon input we added, and the brown rectangle represents the soil, so you can see the green input has been added to the soil. If we go 5 times that rate, what we'd expect is we'd expect 5 times more of the carbon going to the soil. And so we really wanted to test this in relation to the deficit. So essentially, when you increase your input from 1 times to 5 times, you'd expect the ratio of new carbon in your soil to also be 5 times higher. This figure here was what we showed as we looked at this ratio between the 1 and 5 times addition on the y-axis in relation to saturation deficit. The dotted line I've got is this ratio as if there was no effect of deficit. You'd expect that most of the soils would fall out on that just 5 times input. But in reality, what we found is that soils that had the higher saturation deficit or a larger capacity to store additional carbon actually had more of that carbon retained that we added compared to the ones that had lower saturation deficits or were closer to saturation. And so this is still fairly new data and would need to be looked at a lot further. But I think one thing to point out is that when you add carbon to soil, that carbon will continue to decompose and respire. So what this is sort of showing is that soils, when you add, or one thing that's shown is when you add more carbon to soil, a higher input of carbon, the soil responds and you will get more carbon going into that soil. And the other part is that the soils that have the larger deficit are actually looking like they're retaining more of that new carbon. So this is really promising if we, with this knowledge, if we know the soils that have the capacity to increase, it really should, it does support the fact that if we increase inputs to those soils, they should respond more than ones that are saturated. If we move on to the DOC absorption experiment, so this again is the dissolved organic carbon that's sticking to the surfaces. So it's looking at the interaction of your dissolved organic carbon and the mineral surface itself. So this is not a microbial mediated pathway anymore. It's just purely the interaction between the carbon molecule and the surface itself. To do this, what we do is we use a Langmuir plot. So essentially we take a soil and we add different concentrations of dissolved organic carbon to the soil, and we shake it up relatively short term, and then we look at the difference in how much is absorbed onto the surface. And with these Langmuir plots, what you can do is you can extrapolate and work out the maximum amount of carbon that could be absorbed onto that surface based on the response you see. And so in this figure, I've showed kind of the calculation here, and Qmax in the equation refers to this maximum amount of carbon. So on the, just to show this in the figure, on the y-axis we've got the DOC absorbed, and on the x-axis we've got the DOC concentration. So essentially, as you increase the concentration of DOC, you get more absorbed into your surface, and from this information you can work out the maximum amount that can be absorbed. And for this, we wanted to test, does this maximum amount of carbon that can be absorbed on the surface correlate to the predicted deficit we used with that theoretical model. And one thing we did find out was that when we look at the carbon that's in this associated with minerals already, so what we measure, and then what we could additionally absorb on using this DOC absorption experiment. This is represented on the y-axis. So the sum of those two parts in relation to the upper limit, it gets a fairly close, it starts kind of falling out close to the 1-to-1 line. So the dashed line here is the 1-to-1 line. So if both of them, if on the y-axis, both the measured fine fraction carbon or that associated with the minerals and what could be additionally absorbed onto that surfaces summed together, that's very close to what we're predicting with this upper limit. So this, but if you do pass a slight relationship through there, the R squared's pretty poor, but it's approximately about 70% of what you predict with the upper limit. But it is sort of starting to head towards the upper limit, or this 1-to-1 line. So potentially this Qmax or the DOC absorption with what's already in your soil could be used as sort of a proxy to identify what this upper limit could be as well. But also it could be used as maybe a way of looking at what's achievable relative to that theoretical model. Another really interesting part of this DOC absorption work that came out, and this is fairly new, only quite recent, is if you look at the binding affinity. So this is the strength of binding that's required of that organic molecule that's binding onto the soil surface. That strength is actually higher with as you get closer to saturation. So on your y-axis here, we've got this binding affinity, and on the x-axis, we've got the maximum DOC absorption, or that Qmax that we talked about before. As that maximum DOC absorption on the x-axis approaches 0, so you get closer to saturation, the strength of that binding or that binding affinity increases. And so it actually gets a lot harder to get additional carbon onto those surfaces. And so I think this is a really important thing to note, is that I think this kind of starts to suggest the type of carbon will become really important as you get closer to saturation. So when you do have a big capacity, it's really easy to get carbon on, and as you get closer to saturation, that gets a lot harder to get additional carbon, and that's probably things like charge of molecules are really important, and that interaction there is having an effect. Just to summarize so far where we've got to is, we know that mineral surfaces are important, but this is a finite property, and it's an inherent property of soil, so we can't really increase our mineral surface area of a soil. But we also know that increasing carbon input to soil does increase carbon. and that if you have mineral surfaces there and more mineral surfaces, you're able to protect more of that carbon. So if you know the capacity of your surfaces or what's available to store additional carbon, we would get more efficient or faster carbon gain. And that DOC absorption and surface area together, I think, are going to be really useful in identifying these soils that have greatest potential or their ability to store additional carbon. But I guess the question there is like, so what? We've got this information, but how do we actually use it in a useful way that can be beneficial to farmers or people on the land who are actually trying to see the gains? And can this information be used in conjunction with farm scale monitoring? So some farm scale monitoring already happens. If you think of fertility testing for fertilizer, this sort of approach is not really new to, I guess, a farmer. And I guess, can we apply this sort of more fundamental theoretical knowledge to be able to use in more management decisions on day to day? And so this was part of the second bit of the work that we did. What we did here is we tried to translate this work into the farm scale. So I'll just highlight what we did here for this is what we actually went to the Lincoln University Dairy Farm close to Lincoln down here, and we were fortunate enough that a lot of prior knowledge was known about this farm. And one of the things that was known about was the soil type across the farm, and it was well mapped. And so on the left hand side here we've got the farm map with the soil types overlaid. Acknowledging that most farmers probably don't have this information, we did a sampling approach where we did 200 samples across this farm, so it was about 160 hectares, and a balanced approach. That would mean that if you didn't have this prior soil knowledge, you could still do the approach and be okay. Another reason we chose this farm was that it was a relatively high input farm, and because of this range of soil types, it was a good way to test, I guess, our approach based on what we know already, because you could expect to see differences in soil type with deficits and upper limits. But because this farm is close, is a high input farm, it's probably closer to saturation for its soil carbon. Yeah, so on the right hand side here, we've showed the 200 samples, the distribution of the 200 samples across the farm. And when we went to each of these sampling locations, we took soil cores, and then we measured things like soil carbon, bulk density, surface area, all the things we need to do to look at the upper limit and also work out the deficits with the intention of looking at the sequestration potential of this farm. And this is really trying to say, can we take this kind of a larger scale work and bring it down to the farm scale? If we look at the total carbon stock of this farm, so on the left hand side we've still got the soil map, so you can sort of see the differences, and on the right hand side the contour plot, this is total carbon stocks to 15 centimetres, and it's expressed as tonnes per hectare. So you can kind of see the green scale on the bar shows the different stocks, and then the contour plot shows those stocks across the farm. So we used the information we got from individual sampling points and brought that together to make these contour plots. So essentially what we can see is the bottom left hand corner of the farm has higher carbon stocks in relation to the top sort of block of the farm, and the carbon stocks really correspond quite well to soil type. If we look at the surface area, we get a similar approach. So bottom left hand corner, the darker greens have got the higher surface areas, and as you move up the farm you get lower surface areas, and the range of surface area here ranges from about 40 metres square per gram up to just above 65. So we have got a good range of surface areas, but also not the range that we'd see across New Zealand soils. So allophonic soils can get a lot higher surface area. So on other farms we might expect to see a wider range of surface areas as well. When we use this information and translate the surface area into the upper limits, what we see and then work out the deficits is we can start to see that deficit also seems to follow the soil type as well. And here on this farm we have deficits of tonnes per hectare of between sort of 3 and up to about 15 tonnes of carbon. So not huge deficits in this farm, but still sort of room to move. But it's not until we actually look at the proportion of this deficit in relation to its upper limit that we can maybe start to see some nice information. So this next graph here is looking at that deficit as a proportion of its upper limit. And so on this contour plot here, I guess the thing to note is the lighter colour, or the lighter green, is the areas closer to saturation, and the darker green is the areas that have got more potential. So in this farm, the greatest potential to increase carbon would be within the blue kind of box, and the areas closer to saturation would be in the kind of red circle. And so in terms of management, you might say the area in the green circle is, because it's close to saturation, it's really about maintaining carbon stocks in that area. And if you were going to have targeted mitigation practices, you want to apply them in the blue square, essentially. Another aspect of what we did with this work, because we have the soil type map, and we put this into GIS, so we knew the land area under those soil types, we could look at the sequestration potential of each soil type. And we did this in this table here. So we've got, on the far left-hand side, we've got all the different soil types. We know the area under those soil types. We knew the mean carbon stock with each soil type, and also the deficit. So what you can do is you can work out the total farm stock and also the total sequestration potential. And I guess one thing to note here is that you do have quite a range of different carbon sequestration potentials relative to the different soil types. So that could also be a very useful information for management as well. The total sequestration potential for this particular farm, if you sum up all those, was around 1,200 tonnes of carbon, which doesn't seem like a lot. And it's
Hello?
So it's not insignificant, and it does play a small part going towards carbon that is vulnerable to management change. And this is generally a mineralizable fraction that microbes or soil biology can access, and changes in management mean that this fraction is particularly vulnerable to loss, but it's also linked to nutrient supply. And we can measure this fraction by either incubation or by water extraction. And this figure here shows the proportion of that mineralizable fraction of carbon relative to its total carbon. And so that's on the y-axis, and in relation to surface area on the x-axis, we can see that the soils that have high surface area actually have a lower proportion of that mineralizable carbon or vulnerable fraction that can be available for this, or vulnerable to management change, essentially. And this is shown by that red area. So as you increase surface area, that fraction that's mineralizable decreases. And this information was done for a range of pasture soils across New Zealand. But we really wanted to take this and test it at the farm scale as well, to see how does this stack up at the farm scale. So we did a similar approach here, where we looked at this water extractable carbon content as a measure of this vulnerable carbon fraction. And I guess the question here was, would this information be useful for management decisions? So if we look on the far right-hand one, the contour plot, the darker the green means you've got higher proportion or higher amount of that vulnerable fraction. And that's in tonnes. If we look at this now in tonnes per hectare, we've got the range of this vulnerable fraction across the farm is sort of between just under 3 up to about 5 tonnes of carbon. So quite significant amounts of carbon vulnerable to change in management. And so this information I think would be really useful for management decisions, such as if you were choosing whether to have winter grazing or cropping. We might have periods of, like a fallow period, where you've got no carbon inputs going into your soil. So in these instances, the soils that have high amounts of this water extractable carbon or mineralizable carbon might be expected to have greater losses of carbon. So if you knew this information, could you do strategic management decisions to minimize those losses? And so that was quite interesting to see that the distribution of this across the farm. And even though it's sort of run under the same management, you do still get differences in this vulnerable fraction relative to the different soil types. Just moving on beyond beyond this work a little bit more too, thinking about applications at the farm scale with what we know to date, is that we can identify soils that have the greatest ability or increased ability to store carbon based on these deficits and upper limits, and we know that soils with high surface area can hold more carbon. So one of our colleagues, Mikko Kirschbaum at Landcare, has done a scenario recently that's just been accepted in soil biology and biochemistry, where he looked at transferring carbon inputs from, in a hypothetical situation, from a soil that had higher surface area or more mineral surfaces available compared to a sandy soil that had maybe a low surface area. And what he did here is he ran a scenario where he in a modelling sense, that he cut and carried and took all the inputs or the dry matter that's produced from the sandy soil that has low capacity to hold carbon and transferred it to a clay soil that had a higher capacity. And you lost some carbon on the sandy soil, but the gain you got from the increase in the clay soil was larger than the sandy soil loss. And so when you brought that both together, you had an increase of about 4%, or change of your soil carbon content of about 4%. And so you can kind of start with this information. It's quite cool to see that it's sort of been used or applied elsewhere with models to run different scenarios. And I guess the question is, can we then use this information that we know with things like effluent application, or now with nitrogen application within the recent nitrogen caps that have been proposed? And can you basically target our application of nutrients to the areas that have high capacity to get increased gains, but also minimize the losses we might expect? So just onto some future work that I think would be really useful to do would be looking at this farm scale targeted approach. So we've identified areas that for this particular farm that would have the ability to increase carbon, or areas that have probably greater ability to increase carbon. And does that actually translate to, if you increase the inputs in those areas, would you see a response? Another aspect here is understanding the actual limits of carbon input in production. So in all this work, we are constrained by how much carbon we can actually put into the soil or grow, or there is going to be a limit there. So what's actually achievable, and what is that limit? And then translating these mechanisms into models. And so we sort of touched on that with that scenario before that Mikko's done, but I think it'd be really useful to start looking at these mechanisms and putting those into models. And some of this work has started happening in things like the MEMS model. But I think that there's a bit of a gap there to kind of advance knowledge as well. And the other thing is this applying the DOC adsorption as a proxy for carbon sequestration. I think that's there's some real promise there. But to look at that, it really needs testing across different land use and management combinations to seeing if this combination of what's already there plus what can be additionally absorbed on actually works across different land use management combinations. And then finally, just want to acknowledge again the many colleagues and collaborators that was part of this work, and it has been a team effort over many years. And then also thank the NZAGRC for funding this work. I guess without their funding, this work wouldn't have been possible. So thank you.
Thanks, Sam. I'll just wait for it. You can stop sharing now.
Yep.
Yeah, perfect. Brilliant. And thank you very much. That was awesome. And the question and answer panel, you won't have had a chance to have a look at it, has been heating up. And I can see that Mike has been answering a few of the questions. So maybe while Sam's taking a moment to have a rest, Mike, I might pose. There was one question there by Rowena, who really wanted to, could you expand on how New Zealand soil carbon stocks are high by global standards? Is there some sort of global average? And if so, how much higher are New Zealand soil stocks, and why are ours the highest? I think that's a good one to start off the discussion.
Will I pick that one up, Sam?
Yeah, sure. Yep.
Yeah. Okay. Well, there are probably several reasons for it. One is that we have a very high proportion of long-term or continuous pasture soils or land uses in New Zealand, and pastures tend to have high inputs of carbon in the soil. So as a function of that, our soils tend to have higher carbon stocks than certainly other parts of the world where there may be a more continuous arable cropping or mixed pasture and arable cropping, or mixed grassland and arable cropping. Another reason is because we have a very broad range of different mineralogies in New Zealand, and we have a relatively high proportion of allophanic soils, which have a very high capacity to store carbon as well. So that's another reason why some of our soils have seem to store reasonably large amounts of carbon.
Yeah. I also saw a question there by, and obviously, Sam, you spend a lot of time talking about the potential for sequestration, but what's the potential for reversal of carbon from soils managed to increase sequestration? And, you know, obviously there was a question there around carbon accounting, which is more a policy question, but I guess, you know, back to, yes, there's the potential for sequestration, but there also is the there is the potential for loss of carbon as well. Do you want to bring a comment in on that? Or you or Mike, yeah, whoever would like.
I can start and then Mike can finish off, but I think there is definitely that risk of loss of carbon, and I think it's a balance between your inputs and outputs. So anywhere you have, I guess, a lot of outputs going away, you do run the risk of losing carbon from your soils. And so I think that it does become an important consideration that I think we need to be mindful of that, and I think manage soils to maintain stocks of carbon as well. So if we know that soils have got high amounts of carbon and they're close to the upper limit, there is only really one way that they can go, and that is to lose carbon. So I think then it becomes a question of maintaining those levels of carbon, and that's really important. So yeah, I guess this reversal or loss of carbon is possible, and I think that needs to be managed carefully to keep it or maintain it. Do you have anything else to add, Mike?
Yeah, I mean, I think one of the interesting observations from the work we've done so far is that that the soils that have the greatest capacity to store or stabilize carbon are those with a high mineral surface area, and it is those soils which have the lowest mineral surface area that have the greatest risk of carbon losses. So I think it goes to sort of emphasizing what Sam said earlier, and that is around targeting our management practices to increase the carbon inputs to those soils that have the greatest capacity to stabilize additional carbon, and thereby reduce the risk that we have carbon losses from those soils that are relatively, where there's a greater vulnerability to carbon loss.
Thanks both. I see as well there's a couple of questions coming in, and you can see me busy trying to scan through all of the questions that are coming in, so we'll try and get through as many as we can. There's a few on methodology. So Kathy Brown had a question around what methodology was used to map the soil on the farm, so a farmer can use it to change management practices. And then there was another question by Nigel who's sort of related to that. What methods are there to verify, you know, the carbon increase? So, you know, can you comment on the methodology out there at the moment?
So the methodology we used for what we showed for the contour plots was basically like a form of kriging, so looking at how the carbon between, I guess, nearest neighbours changed, and from that you can form these contour plots. And so for this piece of work, it was kind of like a first generation or scoping study to see if it works. And so whether that's the approach you'd use for a farm scale going forward is, I guess, work through, particularly with the number of samples we used as well, to minimise cost. Yeah, I guess on-farm, if you wanted to do this on-farm more practically, I think you're probably going to be limited by cost, and so number of samples available to take, acknowledging the analysis cost in the laboratory is going to limit that. So I think there's probably a bit of work to be needed to be done about how many samples you'd need. And Paul Mudge's report that he did on farm scale monitoring sort of has an aspect of this cost tied into it. And so then it becomes a piece of work for this kind because between the difference between monitoring and what we're doing here to do this upper limit stuff, you might have slight differences in the number of samples you need. So there is some, I guess if this is going to be applied more widely, it would need to be worked out a bit more closely.
Okay, cool. Thanks, Sam. I see as well, there's some questions around Jim Crush's question around most of the sea research seems to be based on that 0 to 30 centimeter profile. What if we develop forages with bigger, deeper roots with more roots greater than 30 centimeters depth? And there's a couple more questions as you go through around, does increasing organic matter change the upper carbon limit? So there's obviously a few people are quite interested in trying to understand the limits that you work at and what that means for soil carbon.
Yeah, I think increasing rooting depth to increase carbon inputs at depth is a great idea, and I guess a lot of our pasture species, like obviously Jim's been doing a lot of work in the breeding of these species, but a lot of our pasture species used in New Zealand are quite shallow rooted. So if we could get deeper rooting species, that would be really helpful, or really important to help increase the input at depth. And in terms of what, and so what was the question about increasing input in relation to upper limit? Is that what was, Sinead?
Oh, there was a question here from Edgar. He said, does increasing organic matter change the upper carbon limit?
Essentially, no, it wouldn't, because in what we looked at, it's related to mineral surfaces. And so mineral surfaces is kind of an inherent property based on your, I guess, your parent material and the weathering process of that parent material. So essentially your surface area is unlikely to change with increasing carbon. And we haven't really seen evidence for that. We have had some work where things like water adsorption does increase when you increase carbon. But in terms of surface area, I don't think that would be hugely affected by increasing carbon.
Yeah.
Maybe I could just add to that, Sinead.
Oh, great. Yeah. Yes, go, Mike.
So, I mean, what Sam said is exactly right. I guess, so we know that carbon is a fixed proportion of soil organic matter, and so definitely increasing soil organic matter content will increase the carbon stored in soils. But of course, going back to the points that Sam made earlier, we know that there is this sort of apparent upper limit to how much carbon can actually be stabilized by the soil. That doesn't mean that we can't increase the carbon, the total carbon in the soil beyond that. It's just that that additional carbon would be relatively unstable and therefore susceptible to loss with disturbance. And then just going back to Jim's question earlier, and you know, Sam's exactly right, and certainly something that's been discussed quite a lot is if we can increase carbon inputs at depth, where we know that where there's a greater capacity, well, there's a greater saturation deficit and a greater capacity to store more carbon, that could well lead to increases in carbon storage or stabilization. I guess the key challenge there is getting enough carbon into those deeper soils. And at the moment, most of our plant species have very concentrated root systems in the top 15 centimeters. Even with deeper rooted plants, can we get enough carbon in there to make a difference? And that's the challenge. And some of those deep rooted species, whether they're contributing much carbon to the soil, that is, those roots are turning over fast enough in order to sort of be an important source of carbon inputs, is a question. And I don't think we know the answer to that yet.
Yeah, and there seems to be as well, there's a couple of questions here, and whether Mike and Sam, you can answer that. It's really around, you know, what about the addition of things like biochar, adding mineral surfaces to soil to increase the potential, you know, the cost and the practicality of that. Is that something you guys could comment on? Because there's been a few questions around that.
Do you want that one, Sam?
Or do you want to comment on it? Yeah.
Well, I can kick off. So without going into a lot of detail, there was several years ago, there was quite a program of research funded by NZAGRC and MPI looking at the potential to use biochar as a way of increasing carbon storage. I mean, I think it's probably best to go away and have a look at the results from that work. But I think the conclusion is that it's a fairly costly exercise, and yet there's still ongoing research looking at the other sort of co-benefits of biochar. I'm not sure that at the moment that's seen as a very viable sort of cost-effective technology, but it is certainly something that people are still very interested in.
Yeah, one thing just to add is here at the centre, we've recently just commissioned a report that will look at biochar and its potential again, you know, in the New Zealand system, because I think there was an acknowledgement that we needed to look at the developments that have happened in the last few years and see how relevant now they are to the New Zealand system. So that, you know, those reports, when available, will be made available via the NZAGRC website. Looking down, sorry, I'm scanning quickly through the questions, and I had one here, and maybe I'll go to the top one actually. From Phil Stevens, he asked a question: do we have any data, longitudinal or snapshot, on soil carbon in New Zealand pastoral or arable lands, which differentiates between the labile and recalcitrant fractions? Do you know the answer to that question?
Well, effectively, Sam has presented some of that work. So the work that we've done looking at the carbon that's associated with the mineral associated carbon, that fine fraction carbon, is what we'd refer to as the stable carbon, and the particulate organic matter or the water soluble carbon would be considered labile carbon or very biologically active carbon. So quite a lot of our work has looked at both of those pools of carbon. And so I think we've got quite a lot of information on that.
Great. So we're now getting to the end. So maybe I'll just end on one last question. It's around, and again, what's the relationship between cropping and cultivation history and sea saturation deficit? This is likely to be the most important farm management practice affecting soil sea levels. Sam, do you want to try and take that's from Alistair?
Yeah. So in the work we published back in 2017, we had an aspect where we brought in, so our upper limit was defined using long-term pastures, and then we brought in soils that were closely paired to some of these sites that were under cropping management, and the deficits of all those cropping sites were lower than under the pasture sense.
Higher, Sam.
Oh, sorry, higher, yeah. So there's more capacity under the cropping soils to increase carbon because they have had historic carbon loss. And so, yeah, it is, that management aspect is quite important.
Yeah.
Yeah.
Okay, cool. Well, sorry, everybody, we've sort of come to the end of our webinar series. I really want to thank Sam and Mike for taking the time out of their day to come and present here. I also want to like to thank all of the audience. I know time is precious, and to come online and to ask all of the questions that you have. Sorry we haven't been able to get through all the questions, but we will work with Mike and Sam to try and get answers to all of the questions and put them up online alongside Sam's presentation, if Sam's okay to share his presentation on our website. Yep. And finally, thank you to Trina and Kate, who have put this State of the Science webinar series together today. And with that, thanks everybody. And yeah, we'll hopefully have some more State of the Science webinar series in the coming months. So watch this space. Thanks, everyone.
Thanks, Sinead.